Good afternoon, everybody. Thank you for coming. It's my pleasure to introduce the graduate student invited colloquium speaker for this semester. Today we have Wanda Strychalski from Case Western Reserve University. Just to give you a little bit of information about her. She received her Bachelor's of Science in Computational Mathematics from Rochester Institute of Technology before going on to earn a PhD in Applied Mathematics from the University of North Carolina, Chapel Hill. And then she moved on to a postdoc at University of California, Davis, before going on to Case Western, where she is currently, and she's been there since 2013. So with that, I'll hand it over to Wanda to talk about 3D cell migration.
Okay. Well, thank you very much to Garrett and Jake and the graduate students for doing such a nice job of organizing my day. I know it's a lot of work to host somebody, and I really appreciate it. It's nice to see you all. This is my first talk in person, and I think a year and a half. So I'm glad to be here and happy to be here. So today I'm going to try to tell you about several different projects. I may run out of time, which is okay. So I'll just do the best I can to kind of give you a sense of the kind of work that I do. And so I will tell you a little bit about my work on single bleb expansion, modeling that and what we can learn from the models. Also some very new work about how cells use blebs to migrate without adhesion in channels. And then also about looking at different modes of cell migration through an elastic ECM network. Okay, so first let's start out with the basics. What are cells? They are the fundamental unit of life. And we have several different types. So there's non-stationary epithelial cells, which could form tissues or protective layers. So say like within a blood vessel or within, your intestines or something like that, or lungs. Then we have motile cells, such as neutrophils. And this one is chasing bacteria. You may have seen this movie before. So it's not a movie, this is just a still. So it's part of your immune system, and it's going to ingest the bacteria, so to protect it from getting infected. And then we also have cells like fish keratocytes, which is the standard sort of cell that's used for models of cell migration, but they also serve biological purposes. In particular, they maintain the tissue within your eye. And I'll talk a little bit more about this later. And then we have cells like pollen, which normally I don't think of as a cell. So meaning that what people get allergies, so the dried hydrated pollen. And so actually they are a cell that contains the male gametes for plant sexual reproduction. So just a very small snapshot of diversity of cells. And so my area is really looking at fluid mechanics and cell biology. And so the first movie that I'm going to play is an amoeba that will give you dysentery. And so you can see that it has this very amoeboid motion. It's migrating on a glass cover slip. And this is the first movie of Blebs, and I'll talk more about defining them later. But you can kind of see there's like fluid flow and motion, and you can kind of see the deformation of the cytoplasm. And then the next movie, actually you have to watch pretty carefully because it happens quickly. So it's going to shoot out a little harpoon. So you can see it right there. I can't access the pointer on my laptop, but I think it'll play again. You can kind of watch this guy. So that's actually a marine plankton, and they use these, so it's a dinoflagellate actually, and they use these harpoons to actually capture prey and eat them. And there's the same organelles that are used for jellyfish to sting you. So they're called nematocysts. And just again, to motivate, like looking at fluid mechanics, this was a simulation for the actual firing of the harpoon, which is called a nematocyst. And so just to see the effects of the fluid mechanics, I'm going to play this movie that shows the fluid and low viscosity versus high viscosity. And so you can see in a very viscous environment that here in the low viscosity, we have a boundary layer near the barb as it reaches the prey. And here there's not really a boundary layer. And you can see that the motion of the barb is actually moving the prey away from it. It's buffeting it away. And so this is a case where we need a certain high acceleration to change the Reynolds number of the system to transition from a very viscous environment because of small length scales to less viscous. And I'm not going to talk about this work today because that's a whole nother sort of the Reynolds number change from, like, I'm basically in cell biology, we're in a highly viscous environment. And so that'll be another talk on another day for looking at non-zero Reynolds number. Okay, in terms of cell motility, why do cells need to move? For many critical cellular functions, such as obtaining nutrients for reproductive purposes, so sperm, for example, that's still cell migration, a different type than I'll be considering, but still important. Wound healing, so fibroblasts will remodel the tissue if there's a wound, and they'll secrete some collagen and reform the matrix. And also for embryonic development, cells need to migrate to certain areas for proper development of an embryo. And then for evil purposes, such as cancer metastasis, we want to understand what's happening in cell migration so that we could prevent it. And here's some movies showing different modes of migration. The first one is the keratocyte that I mentioned earlier. And so you can see that it's just moving, maintaining roughly the same shape. And we see the retrograde flow of actin, which I'll define for you later on. So that's sort of the standard one. The next one, I guess this one's, they're both playing at the same time. So this is a fibroblast migrating in response to platelet-derived growth factor. So normally it's just sitting there, but when it's exposed to the signal, then it starts moving. And this is just showing a signaling protein row A in the cell. And then this is a blebbing cell. So it's a fundulous deep cell in an embryo, and it's using what are called circus blebs. They're called circus because they're round protrusions that move around the periphery of the cell. It is one cell with two nuclei here. So it's not two cells.
Wanda, can I ask? I'm not familiar with the word bleb.
A bleb just means a round cellular protrusion. So it's hard to, I'll show you movies of that later on where I isolate it. So sorry, I'm getting a little bit ahead of myself, but you see a little round. It's basically, think of just the protrusion in this case as a bleb. I think maybe over here, this movie too, just kind of like these round protrusions coming out of the cell. Thank you for stopping me. Okay, so an important feature for the kind of cell migration that I will be discussing is actomyosin contractility. And so the cytoskeleton is made up of actin filaments. So basically, you can think about these guys. So, and these are comprised of these little subunits, and they are coming off and then repolymerizing so that you've got some treadmilling. But for my purposes, we want to think about there's this network of fibers that give the cell its shape, and remodeling of it allows the cell to take on different, change its shape. And so it can take on different orientations. So like a contractile bundle, an isotropic gel-like network. It could have these tight parallel bundles. So we call this, when they're arranged in this way, they're called stress fibers, and they're important for adhesion to the substrate and cell-cell communication. It could have this more isotropic network, which is seen in mostly the cells that I'll be talking about. And these green connectors here are the myosin molecular motors that walk along the fibers, and then they'll slide the actin filaments with respect to one another to generate force within the cell. So myosin motors can cross-link bind and slide along them to generate force. So it's a very dynamic network within the cell that allows for the cell to change shape and form protrusions during migration. Okay, so what are cellular blebs? I've sort of been using the term without defining it. So blebs are characterized, they're spherical membrane protrusions characterized by a separation of the membrane from the cortex. The membrane is in red here, and the cortex is in green. So typically the membrane and the cortex are attached to one another, so you wouldn't be able to see a gap. It's just on the order of 10, 50 nanometers maybe different, so very, very small length scales, whereas this length scale is on the order of 10 microns, a diameter of the cell. So you can see this one might be 20 microns, radius of 10 microns. And so what happens here, a bleb is initiated by a laser ablation of the cortex. So I haven't defined the cortex yet. It is the part of the cytoskeleton, so it forms the shell around the cell. So it's the part of the cytoskeleton that's right next to the cell membrane. Okay, so what happens is they laser ablate here to punch a hole in the cortex, and then the membrane expands. This is from Eva Pauluk's group. She's currently at Cambridge, I believe. Okay, and there's also proteins that link the membrane to the cortex as well. And this is kind of my zoomed in attempt to show the actin fibers with the myosin motors that are cross-linking and then sliding the actin fibers with respect to each other. And then the blue would be the membrane cortex adhesion proteins. And so the scale here is exaggerated. So normally it's much smaller than in the picture. Okay, so the time scale for this is approximately 30 seconds for this cell type. In different cells, they can bleb much faster, such as in dictyostelium. But for the ones I'll be considering is meant to mimic this type of cell. So what happens is over time, the G-actin subunits would diffuse into the bleb and the cortex would reform. Myosin would also reform, and then that's what's causing the bleb to retract again. So the network is reforming and then it's contracting because of the sliding action of myosin. And that can occur on a time scale that's roughly double the expansion time scale. So that's the general idea. Let me know if you have questions on that. Here's another cartoon to show I like this cartoon that shows the life cycle here. So again, you have the actin fibers that form the cortex. And so here, the cortex is still present. That's what this line means. And then there's proteins that link the membrane to the cortex. So then you have the cytosol, which is the liquid part of the cytoplasm, flowing into the bleb, expanding it. Then the actin and myosin and associated proteins diffuse or flow into the bleb, stabilizes it, and then it will retract again because of the molecular motor activity. And there's a couple of different hypotheses about bleb initiation. One is that it's just, it can be initiated by a loss of membrane cortex adhesion, which is what this is showing. And then eventually the cortex would degrade. Another is that you just would do the cortical ablation, which would be like this picture here. So just people aren't really sure about which mechanism. So that's a whole other story, but I have some work on that as well. Okay, so why study blebs? So the main motivation would be cell migration, and blebs are observed during other processes, such as cytokinesis, which is a division of the cytoplasm during mitosis, apoptosis, which is the programmable cell death. So when cells aren't happy, experimentals will say they'll bleb, and that's probably has to do with the amount of myosin that's in the cell. Cell spreading, which is just you take a cell and put it on a cover slip. But my main purpose would be for cell migration. So meaning that it's used as a leading edge protrusion during cell migration. Okay, and the modeling work that I will talk about today. So I'll talk a little bit about motility, but most of it's going to be on looking at single bleb dynamics and understanding the motion of the cytoplasm and how it contributes to looking at the dynamics of bleb expansion. And by the way, this is just a cartoon, or this is from a paper. This is an image of a cell migrating through a three-dimensional collagen network. And it is a cancer cell that was treated with drugs to inhibit its ability to degrade the surrounding matrix. And so they thought that that would prevent the cell from actually migrating, but instead it rounded up and used this kind of bleb-like or amoeboid-like mechanism of cell migration. So cells can switch from the traditional mode, which is similar to the fish keratocyte with the actin flows and protrusions, to the sort of amoeboid mechanism. And this cartoon is showing just a schematic of what this experimental image is showing. So a cell potentially using a bleb to fit in a gap in a three-dimensional environment. And then just to be clear, the little ovals here denote a myosin contraction in the rear. So that's how you can migrate. You could potentially expand the bleb into a gap and then retract your rear, and then you can get translocation in that way. Or that's the hypothesis anyway. So. Okay, so just some experimental motivation, which is why I set out to look at this problem in the first place. So it had to do with this paper from Eva Polluk's group about looking at the role of cortical tension during bleb expansion. In particular, one experiment that they did is they nucleated a primary bleb, which is this first one. And then 5 seconds later, they nucleated a second bleb. at different parts with their different areas of the cell with respect to the first bleb. And they found that the second bleb was always about 30% smaller, regardless of its spatial location with respect to the first bleb. And they said that this indicates that pressure equilibrates fast, at least when it as compared to the time scale of bleb expansion. So the idea is if the cell is pressurized and you bleb the primary bleb, you're going to relieve some pressure within the cell. And so more pressure would be relieved. So this bleb should be bigger here versus here if pressure were not equilibrated. Okay, so basically if you have more pressure relief, this bleb would be smaller, this bleb would be larger. So you have to think about like pressure, that the cell is highly pressurized and this is relieving some. And so what's the pressure going to look like at different parts of the cell? So that was one observation. And then as another part of this work, in terms of using modeling to fit the data, so basically what stops the bleb from growing? So basically if you do like a Laplace law of force balance, the model would predict that the bleb growth is uncontrolled. And so they said that some cytoplasmic elasticity would be necessary to limit the bleb growth because just the surface tension on the membrane could not balance the internal pressure gradient. Okay, and then in terms of another experiment from Guillaume Charras and Tim Mitchinson's lab quite a while ago, they looked at a different cell type and these cells had a lot of blebs in them. And so then what they did is they treated part of the cell with different types of drugs, like drugs that would affect the actin network, so like latrunculin or cytoplasm D, so different treatments that should either freeze the blebbing or cause the actin to depolymerize. And then they also treated with drugs that inhibited, say, myosin to see what would happen. And so basically they saw that in this region where it was treated with the drug, the blebs either froze or retracted. And in other parts of the cell, the cell just continued to bleb normally. And so basically, where it was not treated with the drugs, blebbing was unaffected. And so a result of this paper was that they said, well, hydrostatic pressure does not equilibrate in the cell because basically if it did, then what happens here should affect other parts of the cell as well. And so they said that means that since these blebs, the size and frequency of these blebs were unaffected, that implies that the cytoplasm behaves like a poroelastic material, because that would mean that pressure diffuses slowly through the cytoskeletal network. Okay, and so they propose this model of the cytoplasm has these elastic filaments in it, and when a cell blebs and relieves pressure, that the time scale for pressure to equilibrate is going to be much slower than if it were a viscous fluid. So we have these kind of competing ideas of what happens with pressure inside the cell. Okay, and so some of the questions we want to address with a mathematical model, how does intracellular pressure vary over space and time? Does pressure indeed equilibrate on the time scale of bleb expansion, so about 5 to 30 seconds? And then could we explain how in multi-bleb experiments, you can get that the second bleb is about 30% smaller than the first bleb? Okay, and then how do the material properties of the cytoplasm, so that elastic network within it, how does that affect blebbing dynamics? So the kind of quantities we want to look at is like viscosity of the fluid, permeability, and elasticity. And so I'm going to consider two models of the cytoplasm, so a pure viscous fluid model and a poroelastic model.
Wanda,
Yes.
How do those actin drugs, like latrunculin, inhibit the blebbing? Is it that they dissolve the cortex, or how do they do that?
Let's see. So I think latrunculin freezes the actin network, so then the blebs would be frozen. I have to.
Okay. Well, I thought usually the blebs corresponded to like the membrane tearing off.
Oh, so the cortex will reform and then cause it to retract. And so if you freeze the actin network, then the cortex cannot reform.
Oh, sure.
So yeah, that's a good question.
Okay.
So actually, the latronculin in particular, there's a couple different effects from latronculin. It's not just that latronculin will cause a global and a local effect. So that one's a little bit tricky, but I think it's like rock or something like that. Is the ones that affect myosin actually have the true localized effects. So there's a whole table of, yeah. So I guess I'm leaving it vague on general because there's actually quite a bit of detail in terms of which drugs affect the blebs in different ways. Yes.
Probably silly question, but how universal is this description of the cytoplasm supposed to be? Are these cells supposed to behave in the same way as the cells that you showed before? But
that's a great question. So I was just having a conversation with Shankar about this. So in the paper, so actually your guest, Al Mahadevan, who's coming soon, can talk about this in more detail. So he's the theoretician that was working on this description. So they would say it's a universal description, at least on the time scale of like tens of seconds. So for longer, it's actually going to be poroviscoelastic. So that's because you have turnover of the actin network. So then it becomes more like a mixture of elastic and a fluid. But so on the time scale of tens of seconds, they would say it's quite universal in cell type. But obviously there's a lot of heterogeneity. And if you look at migrating like dictyostelium cells, which is they're slime mold cells and their motion can be very amoeboid, I would say that this poroelastic description may not be the best for them. Yeah, and so depending on how far I get into the presentation, they do propose a universal scaling law, which I don't see the exact same thing. So I would say that yes, it's not clear that a poroelastic description would be applicable to every cell type. Any other questions? Feel free to stop me at any time. So I'm not going to go into too much details about the model schematic, but I'll flash some equations on there just to convince you that I'm a mathematician, I guess. But the model consists of membrane, cortex, and cytoskeleton as elastic objects immersed in fluid. I'm going to assume Stokes equations for the fluid equations. So just the fluid equations with no inertia. And the motivation is that the length scales are quite small for cells, so you don't have those inertial effects. The cortex and the cytoskeleton are treated as permeable materials, whereas the membrane is modeled as impermeable. And I just have some membrane cortex adhesion modeled by elastic springs. So there is fluid on the inside and the outside of the cell. And I use the immersed boundary method to simulate fluid structure interaction. I see a question in the chat, but I don't.
That was actually me sending
Okay.
something out saying if anyone has questions, just to unmute.
Okay, all right, thanks.
All right.
Because I can't even see my mouse right now. On Zoom, it's like a 50-50 chance whether I can see my mouse. Okay, and so the immersed boundary method just allows you to simulate. So if you've got a structure that's exerts elastic forces, then those forces are basically convolved in a way that they exert forces onto the fluid. So it lets you convert sort of between coordinate systems. So the model is put into this like sort of particle tracking Lagrangian form, but the fluid equations are simulated on an Eulerian grid. So I don't want to say too much about that. So here's what the equations look like. So the little f's denote force densities on the fluid grid, which I'm not showing. So we've got force balance on the fluid. This just says that the area or volume is conserved within the cell. And then we have additional force balances on the cortex and cytoskeleton. Because there's a porous slip velocity, the drag force is a constant times the difference between the fluid velocity, which is this F star U, and the velocity of the structures. So you can use these force balances to update you can then find the velocity of the structures based on the force balance here, and you can just update them with like a forward Euler update. And so there's no motion unless you induce a bleb, which I do by removing some links, and I'll talk a little bit about bleb initiation.
That cyto drag there, let's see, I'm thinking that's a force of the cytoskeleton, onto the to the water or?
Yes, so the drag force, so it doesn't move exactly with the fluid velocity, it moves with the slip to it, yeah.
So you think of the cytoskeleton there as being more internal and the cortex being at the edge?
Yeah, so one could model the cytoskeleton as one structure with the cortex. I just decided to separate them out. So, and that's more for, I want to compare the difference of having it versus not having it. And I do have little springs connecting the cortex to the cytoskeleton as well, which is what this adhesion force is here, but that it's, basically like they're on top of each other. So that's why I didn't include that in the model schematic. So the most important adhesion or attachment forces are between the membrane and the cortex, because that's what I removed to initiate a bleb. So I'm kind of glossing over the model to focus on the results. And so the idea here is that if you are just a quiescent cell, and these cells that I'm thinking of are more with the movie of the cortical ablation. So they're around and then you punch a hole in the cortex and then it expands. So what I do is, so there's actomyosin contractility on the cortex, which is transmitted to the membrane through adhesion. So this yellow arrows denote tension. The white arrows are the normal forces as a result of that tension. So the cell is very pressurized. So there's high pressure on the inside, low pressure on the outside. And so what I do is I remove some of the membrane cortex adhesion links, and then that force from the actomyosin contractility is not transmitted out to the membrane. And so you get a pressure gradient, which then leads the membrane to expand and the bleb to form. So that's the picture that we want to keep in mind. And then here's a comparison of with or without the cytoskeleton. So the viscous fluid cytoplasm versus the poroelastic. And so you can see there's quite a bit more pressure relieved in the poroelastic because the cytoskeleton is being compressed as the bleb expands because the area within the cell is being conserved. And since the color bar kind of decreases quite a bit, let me kind of zoom in there just to show you some details here. So this is with the fluid cytoplasm, and we see this very step-like behavior in intracellular pressure. And so you can see it's very low within the bleb, it fills in. And some of the papers in this area say that like if you have a fluid cytoplasm, that pressure equilibrates instantaneously, which is not true. It is a faster time scale, but it's not instantaneous. But we see this very different like step-like profile versus the poroelastic, where we basically see once the bleb expands, there's like a diffusion of pressure across the cell, and then it globally decreases over time. Sort of two time scales, there's the propagation across the cell and then the decrease, basically the rear kind of decreases and there's a global decrease. Okay, so the whole point of this was to look at multiple bleb experiments. And so I'm gonna just try to mimic the experiments by initiating blebs. One at 90 degrees, the other at 180 degrees. And so you can kind of see that the bleb expands and then things equilibrate. And if you think about it just from like a physics point of view, if the blebs have the same like neck size. So here, if this distance where I remove the links is the same as that distance, it makes sense that eventually they would equilibrate to the same size. Okay. But what you actually see here is in terms of bleb size. So the first one actually increases, and then as the second one relieves pressure, the first one actually will just decrease in size. So this is not what they see experimentally. Okay. So this does not seem to match what we were looking for, which is that the second bleb was 30% smaller than the first bleb. Okay, so now let's just focus a little bit on the poroelastic model. And so one thing that we could do with the dynamic model was to just try to understand the effects of the poroelasticity, including that into the model. And so what I did is I basically did a parameter study where I varied the stiffness or the bulk modulus of the cytoplasm and the permeability of the network. And just going back to the Schross paper that I commented on before, where they were treating a blebbing cell with drugs in different part of the cell, they said that in their poroelastic description, that this was there's a diffusion coefficient for pore-elastic material. And so then if you look at the scaling, so they used a scaling argument to talk about the characteristic length, but then you could just change it to an argument about the characteristic time for a bleb to expand. So the characteristic length would be like the diameter of a cell. Okay, so when I did simulations varying the permeability and the stiffness, what's nice is that they seem to collapse onto a single curve if you multiply the bleb expansion time times the stiffness of the network. And so it seems to follow power law, like the permeability to minus 0.9, which seems consistent with the scaling argument. However, I did do 3D simulations, and I'm not going to present too many results from them today because they're very similar to the 2D. But one difference would be the scaling argument. So the scaling appears to actually decrease in the 3D. I could not get a nice collapse of the curves. So it's something like kappa to the minus 0.7. Can't explain that, but this is part of the reason why we do these simulations, to try to have maybe question some of the scaling arguments and delve a little bit deeper into them. Okay, so now let's do multiple blebs with pore-elastic model. And again, the pressure decreases quite a bit, so we're gonna have to adjust the color bar to see what's happening. And the one thing I can do with the simulation that they can't do with the experiments is that I can run it out for a really long period of time. So in the experiments, the cortex will reform. Okay, and then the bleb's gonna retract. All right, so now we see some nice colors here. And so what we see with the 180 degrees is that you can see there's some differences in the pressure, intracellular pressure for the 180 degrees versus 90 degrees. So there seems to be, these color bars match across each column. And so we can see that there seems to be a bit more pressure relief with the 180 degrees. So let's try to quantify this to see what's happening. So this is kind of a summary of, so we've got 3 different values of the permeability. Okay, so if the permeability is high, that means water can flow through the network more quickly. Okay, and so what happens there, we kind of see what we saw with the fluid model, where the first bleb expands and then it kind of shrinks as the second bleb expands, and then they appear to approach the same value. So this is more fluid-like behavior. And we do see a little bit difference. The green and the blue indicate whether it's a 180 degree bleb or a 90 degree bleb. Okay, and on the other extreme would be very low permeability. So water it flows through the network much slower. And so what we see is that there's not a huge difference between the second bleb size and the first bleb size. And this is the ratio of the bleb sizes, second to first. So if it were 1, they'd be the same size. And so over here, it's on its way to 1, basically. And so for this intermediate value of permeability, we see over time that there is a difference maintained between the bleb sizes. And it's sustained for a long period of time. But what we would see if we just ran the simulation out for even 100 more seconds is that we would actually see eventually they do all approach 1. So the relevant time scale biologically would be 30 seconds. And by the way, this black line would be the bleb size. That's for 1 bleb. And so we can just take a look at the pressure at 30 seconds, which would be the relevant time scale. And so here they're both kind of small. Here you can see that the top one is smaller than the side one, this is kind of what we're looking for. A noticeable difference between the second bleb and the first bleb. And this one, this is out of equilibrium. So it's hard to say, but you would see that the first bleb contracting in, which we don't see. Okay, so just to summarize the 2 bleb simulations. So if each whole size or bleb initiation, neck, so if the whole nucleation sizes are the same, eventually the blebs will reach the same size. So the second bleb is always smaller than the first bleb on the time scale of 30 seconds, as long as that second bleb is initiated with that time delay of 5 seconds. And what we see for our initial, on the time scale of 5 to 30 seconds, most of the bleb expands and we see the pressure is actually not equilibrated. And that time scale for pressure equilibration is determined by pore elasticity. Okay. So basically we would say that both papers are right. So the pore elasticity and the fact that pressure is out of equilibrium can actually explain the example or explain the experimental results that Ava's group saw. And so basically we do think that pore elasticity is important for determining bleb size and explaining those experiments.
So are they able to experimentally watch these longer? I mean, in the sense that if they could, would you expect the blebs to get to the same size?
If they could somehow prevent the actin network from reforming, that's one possibility. Another possibility is that you actually you could get uncontrolled bleb growth without the cortex. So basically the idea that, so if the cytoplasm is truly pore elastic, then you should be able to limit the bleb size without, like if you didn't have a cortex there, then bleb size should be limited. But if the cell is highly pressurized, then you might actually get just uncontrolled growth.
Turn yourself inside out.
Yes, but I don't know if one can do that, like locally treat the cell like that. It might actually affect other parts of the cell as well. So it's hard to tease that out, which is one good thing about mathematical modeling or computational modeling. Yes.
So when a single bleb is created, is there a simple way of understanding how much the pressure in the cell is reduced? I mean, there's a certain fractional volume increase. Is the change in the pressure related to that in any way?
Yes. So if you look at the Tenevez paper, they have a nice force balance. And so from their model, you should be able to get an idea of the pressure relief. I don't know it off the top of my head right now, but yeah, so people have done like more simplified, like modified Laplace law models to look at the equilibrium pressure. And actually I should look into that for the 3D. So the force balance models they would be applicable to the 3D model, which I do have, and I could look at the pressure relief compared to theirs. Yes.
Does a bleb ever rupture?
In my models, yes. Yeah, so it's pretty easy to get uncontrolled bleb growth in my models. And all you do is if you do the fluid, for the fluid cytoplasm model, if you just decrease the membrane stiffness and surface tension term, then you can get like a huge bleb. And so I comment on that in the paper. I think I might have a simulation of that.
Has it been seen?
I don't know if experimentally it's been seen. It could be maybe during like what's called cell necrosis. You could have that happen. I don't recall reading about that actually, but I mean, it makes sense, right? It's kind of like a balloon bursting, right? So I would think it could happen. Okay, so as part of this paper too, we looked at kind of a reduced model where if you look at the network displacement as following this, so this would be like this is the stiffness. So this is a model for displacement of the network. So it's a diffusion equation. And basically if you apply, if you have a network and then you remove one end of the network and just basically let it contract, which is what this boundary condition is doing, then you can derive an analytical solution using Fourier series. So just trying to understand this idea of the timescales of pressure propagation. And what you can see is that if you look at the force on the wall, on a short time scale, it does experience a drop. And so you want to think about this as like there's Fourier modes and there's wavelengths. And the scaling arguments are really focused on the dominant wave number, and there's contributions from the other terms as well. So basically that you can experience a force against the wall on a short time scale, but then there's a long time scale for the system to equilibrate. So it's not just one time scale, there's multiple time scales. Okay, so I definitely will not get through everything, but another interesting feature of blebs is looking at the architecture of the cortex. Okay, and so this is a scanning EM micrograph showing the structure of the cortex. So we can see that it has this like network, you know, isotropic sort of network. Looks like a big net maybe, the cell. And so one question is, what happens to the cortex during bleb nucleation? And so these are just some experimental images from some colleagues at Hunter College in New York, and what they saw in dictyostelium. So you can see this is where the bleb's going to grow. After a few seconds, it grows. They actually see these like holes form in the cortical network. And so the idea is, how do you get holes in an actomyosin meshwork? And so just for fun, I had some students use the cytosm simulation, which is agent-based model. So a bunch of like actin fibers that can cross-link because of so it's you have actin, myosin, and cross-linking proteins. And so you can just simulate things very easily and vary parameters. So I'll just show some cool movies from that. So we basically took some, you know, parameters from the literature, and there are little arrows on this to indicate the directionality of the actin filament. And so actually I'll play both movies because I think they take a little while to run. And so what we see, you just see a bunch of fibers like moving around. Myosin is in red. And we can see, you know, some kind of action occurring. You see some vorticity start to develop, so some local rotation. And then, you know, you kind of can see the fibers getting aligned. It's kind of hypnotic to look at. So, and then eventually what you're gonna see is, so this one's already has its hole developing. Down here, it's periodic boundary conditions. So you can see kind of like a nice hole forming here. So that's kind of cool, right? So we see that we can get holes just with some actin fibers and myosin. And the idea was like, okay, well, our cross-linkers are going to prevent this from happening. So cross-linkers would just be binding actin fibers or actin filaments with respect to each other, but not moving, like the myosin is actually sliding the fibers. And so we do see this kind of hole and some, you can see the myosin kind of moving around the hole. So this could be a potential mechanism of how the hole forms. So basically this, you know, we've just done some simulations that are just, this is not my area of expertise, so I'm just getting into it a little bit. But diffusion coefficient is important. A lot of the parameters having to do with myosin are important, like the binding rates, the rate at which it binds to actin, the unbinding rate. So myosin has to be, it has to walk along the actin for a certain amount of time in order to align the fibers to get the hole. And then we do need the speed, the maximum speed that it walks to not be too large or too small. Also, the length of actin is important. So you need your fibers to not be too long or too short with respect to the domain size. So just trying to, this is, you know, relatively new work. So just trying to get a sense of what's happening in terms of getting holes in the cortex. So to switch gears, I don't have a lot of time left, so I'll just kind of briefly talk about this. And then there's another part, but maybe I'll just flash the reference up for that. So in terms of three-dimensional migration, so one system that people look at would be this like confined migration. And this is showing actin here, and then this is showing myosin. And this cell does use blebs. This is that same cancer cell that was migrating through the extracellular matrix from before. And so you can see that, so there's blebs and there's also like rear contraction here. And so there's kind of like two ideas of how a cell moves under confinement. One is the cortical flow model. Basically, so you would have adhesion to your substrate, and then you would also have actin flowing along the sides. And then that's the model used in this paper, also from Ava Pollock's lab. And then the other model would be, okay, so the cell is somehow like pushing against the wall and what they call chimning, like a rock climber, kind of chimning on the side. And so we wanted to explore some of these ideas since we built this framework for blebbing. Oh, I didn't realize I had that movie. So that's kind of nice to watch. See the cell kind of moving through the channel. See it kind of blebbing. Okay, so in terms of modifying the models, we do have to add in a channel wall. And I had bleb expansion but not retraction. So basically we added in equations for the density of the cortex, just simple ODEs. So we have old cortex and new cortex. And so the values of the density are constant along these. And then the elasticity for the cortex is just multiplied by the density. And so you can see bleb expands and then retracts. And then this is the density of the cortex, so low for the new, and then it increases over time. And then it will retract once this gets to a certain value. So you have these cycles of expansion, retraction. I'll show you some movies. So this is just a cell expanding and retracting using a bleb. And so it's visualizing the pressure and the velocity. And so we varied the bleb sizes. And as a person that studies fluid mechanics, we don't see motion. And this is to be expected because if you've heard of the scallop theorem, so in order to migrate in low Reynolds number flow, you need more than 1 degree of freedom. And so this is essentially like 1 degree of freedom. And so you think about a scallop going like this and migrating by pushing fluid. So you can't do that in Stokes flow. You can open, if you open and close, you're going to end up exactly where you were. And so this blebbing action is essentially like a scallop. So you have to introduce some kind of other degree of freedom to get motion. And so we tried, okay, let's do an asymmetric channel wall. And so what we see is that we can get some motion, but not a whole lot through cycles of bleb expansion or retraction. The same with adding more asymmetry. A little bit of motion, but not too much. And so I don't have the results yet because this is still sort of like a new realization. But what we see is that with just cycles of blebbing, you need some asymmetry to get a little bit of motion, but you can't get a whole lot. And so we thought, let's add in that rear contraction to simulate the actomyosin contractility. And when we do that, what we see is if you model the cortex as elastic, so if you add a retraction and remove it, then what happens is that you can't go anywhere either. You apply a retraction, and then when you release it, you're just going to end up exactly where you were. So you cannot migrate with rear retraction and blebbing. But if we use a viscoelastic model for the cortex, then we actually can migrate without adhesion. And so that was the key to getting net motion, net forward motion, at least for straight channel walls or asymmetric channel walls. And so currently we're just kind of going through and trying to understand like how much motion we can get through this viscoelastic cortex with retraction and blebbing. Okay, so I will probably stop there. I'll just show you a couple cool movies of mesenchymal and amoeboid type motion. Because it's kind of cool. You can kind of see the rounding of the cell here, and this is through the 3-dimensional matrix. This is the same figure that I showed earlier, but just showing the movies. And another sort of model involves just looking through, like, this is again fluid structure interaction. I'll just show you some movies of the cell migrating through generating protrusions onto an elastic ECM network and moving in. This is more like a mesenchymal model for 3D migration. And then this is all done immersed in fluid. And then I'll show you the rear squeezing. So a cell kind of squeezing itself through a gap by using 2 protrusions. And so we got some results from this, but I'll just kind of show you, or just kind of go to the last slide for my acknowledgements, because I don't want to rush through that. So that'll be another day, I guess, another talk. So the blebbing work was done with Bob Guy from UC Davis, started at my postdoc and continued on. Ava Pawluk also provided, she let me visit her lab a couple of times to pick her brain. Alex also helped because he was at UC Davis at the time. The blebbing under confinement was done with Kalina. And the third project was mostly done with Andre and a couple of students who are working on the cytosem simulations. And thank you for the invitation to come here and for your attention.
In that the last example with the blebber and the channel, the blebs were always coming on the same side.
Right.
Right? And do cells have a memory that they tend to keep blebbing at the same place?
Yes. So you can, because the cortex is weak in there, then it's more likely to rupture again. So yeah, so different people have done models where they allow the cell to kind of bleb anywhere. And so I just haven't built in that capability into the model. So, but yeah, so I'm forcing it to only bleb on the one side at this point in time. But biologically, yes, if you blebbed one place, you're likely to bleb there again.
Okay. And then I'm wondering, if you allow it to bleb and then you allow the actin to reform quickly and stabilize that new bleb, I mean, wouldn't that eventually have to lead to directed motion?
You need adhesion or something. You mean like with no myosin?
Well, yeah, I was thinking as an alternative to the myosin that you could do it with actin polymerization instead. That the actin would flow in there and kind of, I guess it would have to lock it to the substrate, but that's certainly plausible.
Yes, and that, yeah, so that's, I think, what, if you're doing like blebbing-based motility in 2D, you would have to have adhesion to the substrate. So yes, you could bleb, actin stabilizes, and I think that's what's actually happening in dictyostelium.
Okay.
Yeah.
You're saying in this case, you don't actually need to be attached to the substrate.
Right, so I forgot to emphasize that. So we specifically wanted to look at how can we move without adhesion. And so, yes, we could add adhesion into it. And what's cool is that we do see the cortical flow. So if you treated the cortex as like just a fluid and you apply that rear contraction, you will get motion. So you just need, the cortex has to forget its like reference configuration it has to be free to flow a little bit, which makes it numerically really challenging. So if you're trying to do a Lagrangian simulation, but I think we know how to do it, at least in a hand wavy way. So
I think you said it was not pure, but those simulations that you showed with the strings, the two panels, you had that network of strings or like filaments. What is can you explain a bit more? What's the dynamics? What are the interactions? Yeah, I think.
yeah. So you mean you're talking about the cytosm stuff?
Yeah,
these guys.
So you have these filaments, right? And they interact.
The filaments interact. So so it follows a Langevin equation. So they're vibrating just because of like Brownian motion, and then they can bend. They're a little bit bendy and stretchy.
So they have a tension. You have a string with a tension, right?
Yes, and they can cross-link. So if I have cross-link, so myosin, which is the red, can actually bind. They can cross-link one to the other. So like attach a spring between two.
All right. So the difference between one panel and the other is that when two of these strings come together, they either interact or they don't, or they just cross without interaction.
So they interact through the myosin, which will like cross-link them, and then they'll start to walk. So it's like two heads, and then they will walk in different directions towards the plus end of the fiber. So they're oriented fibers. And so they'll cross-link and then they'll start to like slide apart. But if you have the blue, then they'll just cross-link. So if you didn't have the red or the blue, you just have a bunch of white fibers kind of like moving around freely. Does that make sense? So I think I might have the equation somewhere down here, in a different presentation. I was kind of looking at a bunch of presentations here. Maybe I don't have it here, but I have a poster from this one might actually have it here. So these are this is kind of like a little schematic. Oh, I don't want to do that. I wanted to zoom in, but may not be possible. So these are kind of the equations where the x's represent little, like the fibers kind of discretized into these nodes. And so you can have this cross-linking and you can have motion because of a molecular motor, and you can have different, they're diffusing, and then it's just, there's Brownian motion. So it's just Langevin, and the forces are due to cross-linking and elasticity. So it's just like over-damped motion. So I call this an agent-based simulation because it's interacting particles basically. So I did not write this code. That's just a package anyone can download, play around with. try to see the patterns. And that part of the challenge of this is actually like getting useful information out of these. So, and that's something that I've been trying to look at things like volume fraction and like alignments. So it's a little bit tricky. I'm open to suggestions.
Okay. Are there any questions from the online folks?
Could I ask one?
Yes.
Really nice talk. Thank you so much. Does bulk cytoplasmic flow play any role in any of the bleb dynamics? Because this is something that people have, sort of like, or like cytoplasmic streaming, or there's all these phenomena about how the cytoplasm itself sort of often undergoes oriented flows in a lot of cell types. Do you know, have you ever looked at that?
Right, so actually there is a bulk cytoplasmic flow, and I just didn't show the velocity vectors. So there is a bulk flow that's moving from left to right. And so you can kind of see that if you look at the movies of the confined motility, or actually maybe even in, well, I didn't really talk about that, but I can just kind of show you this picture here, which is nice. So for this cell here that's moving through these different mechanisms, so the arrows do indicate the velocity, so that's inside the cell. So the cell is moving from left to right. So there is flow moving from left to right. And what's interesting just about these results is that for the top cell, you actually have an adverse pressure gradient, whereas the bottom one, you have pressure from low, starting out high in the rear and low in the front, which is what you would expect for fluid flow. So yes, I mean, the answer is yes, there is bulk cytoplasmic flow, and you can see it best in the confined motility, which is here. I don't know if I can make it. Oh, that one doesn't have the arrows in it. So all these you can get the, does this one have arrows? Yeah. So this one, the arrows are kind of, this is the fluid velocity, and I'll make better movies in the future, but they're pointing this way, and then as the bleb retracts, they're actually going back in. So you can see that the streaming is going from left to right and then right to left through the blebbing cycles. So yeah, if you don't have fluid flow, then the membrane is not going to expand in this model. Also focused on pressure because that's what the papers are all commenting on, but yes, you can extract information about velocity as well. And forces too.
Anyone ever put any little markers inside the cell to monitor that velocity?
That is a great idea, and that's easily done. Yeah, I should do that. I will do that for this paper. So little tracer particles. Yeah.
Like experimentally, can they do that?
Yeah, they can. Yeah. Yes.
So you show that for one bleb, the cell might move like this, but what happens when there are multiple blebs, and maybe the cell would move and move back like Brownier, or if the cell blebs stochastically, would the, so for the general or for the big picture for the of the cell moving, would it be like a Brownier or like a random walk? So I mean, for in situations where there are multiple blebs, what is the big picture for the migration of the cell?
So one of the sort of big pictures is that for certain type of like an embryogenesis for zebrafish at least, so blebbing cells will actually, the cells will push off of each other in a way using blebs. So that's, in order to migrate effectively, you actually need multiple cells blebbing in order to move like an entire like tissue forward. So there's a paper about that. And so the idea with blebbing is that it's not as persistent as like regular modes of cell migration, like the mesenchymal with actin protrusion, adhesion and retraction models, like the fish keratocyte type of picture. But the idea is that it's less like chemically regulated and more like a physical process, so that it's kind of evolutionarily speaking, easier for the cell to do. So you get less persistent motion, but you get more motion, and then if you kind of push off of each other, then you can effectively migrate. So in terms of like stochastic blebbing, yeah, I mean, I'm forcing it to do this, but biologically speaking, it is. So this gets into the bleb initiation hypothesis. So like, what determines when those attachments detach? And so some people have done detailed modeling of just the membrane cortex adhesion dynamics in terms of like, what's a critical, if you've got a bunch of linkers going on and off, like what's a critical value where you can actually get like a bleb to form. So yeah, that's an important aspect of this so I'm not considering those other factors, and they are important. Yes, Jason.
So sorry to go back to the like cross-linker versus no cross-linkers, but could you remind me what was the intuition why there's holes for the no cross-linker case? Like
So there's holes for both. So the one thing we weren't sure about. So yeah, it's interesting. It's just the evolution of the simulations. So I thought maybe that if you had cross-linkers, that could potentially like fix the network so that it wouldn't be able to form enough to get, so the myosin could like align it into these like holes. I thought maybe and there's probably a critical value of cross-linkers where you cannot get holes to form. So if the network is very rigid, then the idea would be like, you can't move things around enough to get a hole. So that was why I thought maybe I haven't done a lot of simulations with the cross-linkers yet, but it was just because biologically speaking, if we think about what that cortical meshwork looks like, it looks like a cross-linked isotropic network. So there's going to be cross-linkers in there. So we know that biologically. So it's trying to, we don't really have just actin and myosin, right? There's a lot of other factors involved. So the idea was just to see like, okay, so if I add in cross-linkers, then will that keep the hole from forming? There's also a question like, how do you control this hole? Like we want it usually to be like at the leading edge of the cell. So I don't know how to control where it is yet.
So with the cross-linkers, it seems to be happening at the boundary. Is that just coincidence?
Yeah.
Or is there a reason for that?
It's just, it's a random number generator to start with. So
But it has nothing to do with the boundary conditions that you've imposed.
no, it's just periodic. So yeah, if you reran the same simulation with a different like
See
seed.
that top bit there
Mhm.
is linked to the bottom.
Yes, exactly.
Gotcha, okay.
Cool. Well, let's thank our speaker again. That's very
Thank you.
interesting.